Okay, very good evening, my dear friends. Today's our eminent resource person, Dr. P.M. Sujatha Madam Garu, Assistant Professor, Department of Botany, Government Degree College for Women Autonomous, Karimnagar. Madam is going to explain about the Anthoceratopsida of Bryophyta. Madam, please welcome and start your session.
Thank you, sir. Good evening to all, my dear students and sir. Last class, we have finished in Bryophyte the first class, that is Anthocerotops, the Marcant in just a minute. Last class, we have finished Marcantia, which comes under liverworts. Today, we'll start with Anthoceros. Today, we'll start with Anthoceros. So Anthoceros, it comes under Anthoceratopsida. The classification Anthoceratopsida is also called as that group. It is called as hornworts. Hornworts, as you are seeing on the thallus, that is thallus lobe in the diagram. You are seeing. Am I audible, sir? Is it audible? Am I audible, sir? It is Anthoceros. Am I audible, students? Is it audible? Is it audible, sir?
Okay, madam.
Sir, shall I start or?
Okay, madam, please proceed. Well, Janavtar.
Sir, sir. So today we are going to start Anthoceratopsida. Under Anthoceratopsida, we are studying Anthoceros. Anthoceratopsida group members, they'll have horn-like structures. So that's why that is a specialty of this group. Anthoceros, the classification. Anthoceros, it comes under the kingdom Plantae. Division is Bryophyta. Class Anthoceratopsida. Order is Anthoceratales. Family is Anthocerataceae and genus is Anthoceros. So it is named after all the suffixes we have to add just after Anthoceros. Next, Anthoceros. First, habitat. And habit, we'll study habitat. As you know, these are the bryophytes. So they'll grow in moist places, wet places, wherever water is available. And their cosmopolitan distribution. Mainly, they'll grow in temperate and tropical regions. Here, more than 200 species have been identified and 25 species has been recorded in India. Mostly, they grow in moist, shady places. They'll grow on the sides of ditches or in moist, hollows, that is, the cracks which are present on the rocks. Few species grow on decaying wood. So in wood also, if cracks are there, water if it is available, then on the decaying wood also, some of the species of Anthoceros they'll grow. Three common Indian species of Anthoceros. One is Anthoceros erectus, second one is Anthoceros crispulus, and another is Anthoceros himalayensis. About the structure now. That is the structure of the plant body, that is the habit. As you know, bryophytes are here anthocerotales, that is Anthocerotopsida group. They'll have flat thallus, like just like Markantia. Thallus plant body that is flat. It will have lobes, but these lobes are not exactly like liver lobes. Here, that demarcation is there. So just lobes are present. This plant body it occurs in moist, shady habitats in subtropical and warm temperate regions. The gametophyte, the plant body, the thallus, is here also dorsiventrally differentiated and they are rosette like. They are somewhat thick. The thallus is thick. Marcantia is somewhat thin, but here it is thick and they are arranged in rosette fashion. That is, lobes are arranged once beside the other in groups. On the ventral surface of the thallus, that is dorsiventrally differentiated. So on the ventral surface, here only smooth wall rhizoids are present and which helps in fixation as well as absorption of water and moisture. Whereas in Marcantia, smooth wall and tuberculate rhizoids are present. Here only smooth wall rhizoids are present. About the scales, scales are absent here. Tuberculate rhizoids are absent. Scales are also absent in Anthoceros. Some species of Anthoceros are unisexual and some other species are bisexual. Means on the same plant body, male and female gametophyte may be born or it may be born on two different thalli. Male plant will produce antheridia, as you know, and female plant will produce archegonium. So the antheridia and archegonia are sunken. That is, they're deeply seated. Whereas in Marcantia, these are produced on a disc-like structure, or that is a antheridiophore, archegoniophore. But in Anthoceros, they are hidden inside the thallus. They are sunken. That is deeply seated. That too on the dorsal surface of the gametophyte. Then numerous sporophytes may develop on the same gametophyte. Sporophyte will develop in place of antheridia after fertilization. So, if more than one antheridia are present, more than one archegonia if they are present, that will develop into the sporophyte after fertilization. In the diagram, upper diagram, you are seeing the actual diagram, the elongated horn-like structures, that is a sporophyte. In the lower diagram, you're seeing just the picture of that, that is on the thallus. Left side, you're seeing so many sporophytes are born, horn-like sporophytes. Right side, same thing. It is shown singly, that is at the tip of the lobes. You're seeing the sporophyte. On the lower side of the thallus, left diagram, you're able to see the rhizoids also. Here the midrib is not clear, whereas in Marcantia, clear-cut midrib will be there, which is absent here. Then about the internal structure, anatomy. So anatomy of the thallus. Here in the left hand side diagram, upper diagram, you're seeing tears of the thallus. Here, that is, here also it is dorsiventrally differentiated. Upper epidermis you're seeing and lower epidermis you're seeing on the lower side, upper diagram. And from the lower epidermis, you're seeing rhizoids are rising, unicellular rhizoids. They are simple rhizoids. They are not tuberculate, only simple rhizoids are present here. So the thallus is not differentiated into photosynthetic zone and storage zone, which is present in Markantia. In Markantia, clear-cut upper photosynthetic zone and lower storage zone is present in between the upper and lower epidermis. Here it is absent. The cells are all similar. All cells are green and contain the chloroplast. Cells means in between the upper epidermis and lower epidermis, the larger cells, polygonal cells you are seeing, they are all similar and there is no intercellular space in between them. They'll have chlorophyll pigments inside this. Chloroplast are present there. Speciality of this Anthoceros is chloroplast are associated with pyrenoids, which is the characteristic feature of algae. In algae only, in the chloroplast, you'll see the pyrenoids. So this is the algal characters. That is, chloroplasts are associated with pyrenoids, which is a unique feature of anthocerotales, which is similar to the algae. And there are no air chambers or pores. Whereas in Markantia, air pores are present, which will enter into the air chamber. Such chambers are absent. No photosynthetic tissue or the filaments. Here, pores are also absent. Here you can see schizogenous cavities filled with mucilage are present. On the lower side, you're seeing some cavities. In the upper diagram, you are able to see some cavities that is just above the lower epidermis. You're seeing some cavities. These are called as, that is, cavities, and in this mucilage cavities they are. They are also called as schizogenous cavities, which are filled with mucilage. These are often inhabited by nostoc, means in this cavity, algae are present, blue-green algae, cyanophys bacteria or the blue-green algae are present, mainly nostoc. In blue-green algae, the algae called nostoc is present here. So, specialty of the nostoc is that it helps in nitrogen fixation, which supplies nitrogen through nitrogen fixation to the host plant. So this is a symbiotic association between Anthoceros bryophyte and nostoc. Lower diagram, same thing you're seeing here. Lower side cyanobacteria, there is a nostoc colonies in the cavities you're seeing. Next, about the reproduction. Reproduction in Anthoceros, it is by various method, vegetative or sexual, that is sexual reproduction also is seen. Here vegetative reproduction. Now we are discussing about vegetative reproduction in Anthoceros. First one is by fragmentation. Fragmentation means if the thallus, if it is broken down by the death and decay of the lower parts, posterior parts of the thallus, then each fragment, each piece can develop into a new plant body of Anthoceros. Second one, by means of tubers. Tubers, they'll store the food material. They develop at the end of the growing seasons. So after the complete vegetative growth, these tuber-like structures, extra food material is stored there and they are developed during unfavorable conditions, which will survive in the soil for many years. So they are perinate and they grow into the next season in the favorable conditions when plenty of water is available, moisture is there, then each tuber can develop into a new plant body of Anthoceros. Third type of vegetative reproduction, gemme. Just like Marcantia. In Marcantia, gamma cups are the distinctive feature. But here, some species of Anthoceros, they will also produce gemme. So, which are borne on short stalk on the dorsal surface and along the margins of thallus. Whereas in Marcantia, they are produced on gamma cups. When these gamma are detached, they germinate into a new plant body of Anthoceros. Lastly, persistent growing apices. Apices means the apex of the thallus, thallus tip. So that by chance, if it is detached, that will survive for many years and that will, under favorable conditions, it will give rise to the plant body of Anthoceros. So during summer, long summer drought, when drought is there, water is not available, then that time disturbers, persistent apices, they'll survive. And after the return of favorable conditions, they'll develop into a new plant body of that Anthoceros species. Next one, sexual reproduction. Sexual reproduction, it takes place by means of development of a Antheridian, that is Antheridian archegonia. So here sexual reproduction is oogamous type. Oogamous means male and female gametes. They are separate. That is male is motile, small, female is egg cell, larger when compared to the anthrozoids, and it is non-motile. So oogamous type of sexual reproduction will be there in Anthoceros. Male sex organs are antheridia and female one are known as archegonia. Each one is known as archegonium and antheridium. Antheridia plural word, archegonia plural. So they may be born on the same plant or different plant also. Many species are monoecious while some are dioecious. Monoecious means on the same plant body, thallus, male and female organs are developed. If they are developed on different plants, then it is known as dioecious. Formation of sex organs are depend upon the specific photoperiod. So it depends upon the external factor, the light to which it is exposed, that is photoperiod, the duration of the light to which it is exposed. Anthoceros being a short day plant, sex organs develop in winter only because water is essential for reproduction for the moment of anthrozoids. So that's why in winter season and then only this reproduction will take place. Sex organs are developed there and reproduction will take place. Antheridia develop much earlier to the archegonia. That is, protandry is seen. This is called as protandry. Development of antheridium earlier to the archegonium is called as protandry. About the antheridia, which is the male reproductive organ. In all bryophytes, antheridia is club shaped. That is, in the first diagram, you're seeing two antheridia in one chamber. So, antheridia, it occurs on the dorsal surface of the thallus. They are sunken, they are deeply seated. Antheridia archegonia, they are not born on the upper surface, that is outside the thallus, but they are hidden inside the thallus on the dorsal side. So also antheridia, it occurs on the dorsal surface of the thallus. They are developed in acropetalous succession inside closed cavities, and these are called as antheridial chamber. In the first diagram, You are able to see the antheridial chamber with a roof, a covering, and inside the chamber, you're seeing two antheridia. Each antheridia structure now. Antheridia is differentiated into a long stalk and a club shaped body. It will have a oval body and a stalk. That together it is called as club shaped body. The stalk is multicellular. In the diagram, clear cut left hand side diagram, you're seeing multicellular stalk. That means it is made up of many cells and it is slender. It consists of four vertical rows of elongated cells. In the diagram, you're seeing the four cells in two rows they are arranged. So four vertical rows. This side only two, back side also two are present. So four vertical rows of elongated cells are present. The cup-shaped body of Antheridium has single-layered sterile jacket. That is a roof we are calling. That will be the sorry. Inside the roof, you are seeing the Antheridia. Each Antheridia is covered by a sterile jacket wall. So which encloses a mass of androcytes. In the box you're seeing small, that is inside the jacket wall. In each antheridia you're seeing small boxes. Each box is called as androcyte, which metamorphosis means it develops. After development it changes into a biflagellate curved antherozoid. In the left, that is left hand side, in the right hand side, the last diagram what you are seeing is the antherozoid. They are also called as sperms. Mature sperm, the upper side you are seeing two long flagella. So they are biflagellate, which will develop from androcytes. These biflagellate antherozoids are developed from androcyte. So many androcytes are developed inside the antheridium. Development of the antheridium now. Each antheridium, it develops from the upper side of the thallus. One of the cell of the thallus, it behaves as an apical cell. The upper cell is for our convenience we are calling it as apical cell. This cell, it divides transversely into two cells. The upper cell is called as outer cell and lower cell is called as inner cell. The inner cell is the antheridial initial, which that means the starting cell from which complete antheridia will develop. The outer cell, it divides vertically and horizontally. That is periclinal division and anticlinal division, and produces four cells are produced. Afterwards, it divides and re-divides periclinally and produces the wall. That is a roof cells. These are known as roof cells. These roof cells will produce the antheridial chamber wall. That is a roof. These roof initials are developed inside is that is antheridial initial is present. In between them gap will be formed. That will be the antheridial chamber. This roof cells will produce the roof of the antheridial chamber, and now the initial inner shell, that is the antheridial initial cell, it divides vertically and produces two cells, two initials. Each initial will develop into a antheridium. One antheridium. If three are present, three it will be developed. Mostly two antheridia are seen most of the species. That antheridial initial cell, it divides. The lower cell is called as stalk initial, the upper cell antheridial proper, that is which divides vertically and transversely and produces four cells initially, and the lower cell stalk initial will divide vertically and transversely and produces two or more times also. Then it produces the stalk of the antheridium. and blue color line left hand side. From the lower side, you're seeing second diagram. That blue colored line, that is a chamber of antheridium, in which one antheridia is shown there. So afterwards, this primary and androgonial cells, or the antheridial cells, they develop, they divide and redivide mitotically and produces a mass of cells. These are known as antheridial cells, or the androcyte cells. They are the androcyte cells. So more than one antheridia may be developed in each chamber. Next, archegonia. Archegonia is a flask-shaped structure with a long venter, that is, a round venter at the base and an elongated neck at the tip of the neck. Neck canal cells, at the tip of the neck, you will see cover cells or lid cells are present. So about the archegonia. Archegonia, they are embedded on the dorsal surface of the thallus. These are also produced in the dorsal surface of the thallus. They are developed in acropetalous succession near growing points. Toward the tip of the thallus, only they'll grow. The archegonial chambers are absent here, whereas in Antheridia, chambers are present, archegonial chambers, but here at one place, one archegonia is only present, so there is no chamber here. There is no archegonial chamber. Archegonia is a flask-shaped structure. As I said, it consists of neck and venter. It contains, inside the neck, you will see here 4 to 6 neck canal cells, and in the venter you can see, which is a round structure basal part, there you can see venter canal cell, VCC it is called, and below that venter canal cell you will see the egg cell. In the diagram you are seeing the egg cell. Above that venter canal cell, that is the venter, in the venter region, single venter canal cell, and above that long elongated neck is present. In the neck, 4 to 6 neck canal cells are present. Above the neck, you'll see the cover cells or lid cells. That is the structure of archegonia. Now, development of archegonia. Archegonia develops from a single initial cell, that is called as, initial means the starting cell, archegonial initial. So toward the dorsal side, one of the cell, it acts as a archegonial initial cell. This archegonial initial cell, it undergoes periclinal divisions toward the periphery. So it produces 3 layers of cells, that is the jacket initials. 3 jacket initials are produced. When these 3 periclinal divisions are taking place, at the center, one cell is formed, that is called as primary axial cell, or it is a central cell. This primary axial cell afterwards develop into the neck canal cells, venter canal cells, whereas the jacket cells will develop into the archegonia wall. So now coming back to this periclinal jacket initials, that will develop into neck walls, as that is a vertical division, mitotic division, it takes place, and due to that 3 vertical rows of cells are formed that will produce the neck region, and lower one, it produces the venter, that is venter primary. On the lower side, from the central cell, primary ventral cell is formed, and upper cell will give rise to the neck initials. Neck initials, they'll undergo transverse division 3 to 4 times. Due to that, 4 to 6 neck canal cells are produced from the axial cell, the starting cell. So finally, in the primary ventral cell, it also divides and produces the beta canal cell and egg cell. So what is seen in the archegonia means archegonia, a flask-shaped structure with a swollen base, in the venter, egg cell, lower side, above that, venter canal cell. In the neck region, elongated neck region, 4 to 6 neck canal cells, which are surrounded by jacket wall, that is the outer wall, that is periclinal jacket initials, which are arranged in 3 to 6 rows. Then fertilization. Fertilization, it is nothing but the fusion of male and female gametes. Antheridia will produce so many anthrozoids or the sperm cells, which are biflagellate. Right hand side, top diagram, you're seeing the anthrozoids. So the anthrozoids swim in water. Whenever water is available, that time only this can swim. So in groups, in masses, they are produced from antheridia. Some of these will reach mature archegonia. These anthrozoids are produced en masses, in groups they are produced. One of the anthrozoids so many anthrozoids will reach the archegonia, but one of the anthrozoid only will enter into the archegonia by the that is by the secretions of antheridia that is attracted, and it will enter there. When it will enter means a mature archegonia is characterized by mucilaginous mound at its apex. So when the ventacanal cell, neck canal cell, they dissolve and they'll produce a mucilaginous substance. Due to that, lid cells will separate, the cover cells will separate, and it produces a liquid, that is chemotactic movement. Due to that, anthrozoids are attracted towards the neck of the archegonia. Only single anthrozoid, it enters into the archegonium, and it fuses with the egg cell, and due to that, fertilization takes place and zygote formation takes place. Zygote is always diploid. So this is the sporophytic phase. Zygote onwards is sporophytic phase of an that is Anthoceros. Zygote, it develops into the sporophyte. You're seeing in the diagram, in the archegonia, by the fusion of anthrozoids and egg cell, zygote is formed. That zygote develop into the sporophyte. Sporophyte is differentiated into three parts. Foot, here seta is absent. Instead of seta, intermediate cells or intercalary zone is present, which is made up of meristematic cells. Intercalary meristem also it is called. And the third part is capsule. So instead of seta here, intercalary meristem is present. That is the speciality of Anthoceros. Continuously it produces new cells toward the upper side. So about the foot. Foot is the basal part. In the archegonia, that foot is developed. So it is a basal bulbous parenchymatous structure found deeply embedded in the gametophyte. It helps in attaching the sporophyte to the gametophyte. Food structure, just it helps in fixation, and it also helps in absorption of water and nutrients from the gametophyte. Here the sporophyte is dependent on gametophytes for its survival. Next part is the middle part, that is the intermediate part called intercalary zone or meristematic zone. A narrow zone of meristematic cells located between the basal foot and upper capsule. So upper part is known as much elongated horn-like structure, that is the capsular part. Largest part is the capsular part. So this intermediate zone, these cells help in the continuous growth of the sporophyte because they are made up of meristematic cells. They have the capacity of division continuously. Last part, third part, largest part, horn-like, which is seen outside, that is capsule. It is a fertile, major, and conspicuous part of the sporophyte. It is clearly seen, which is long and cylindrical. It is green when young but turns gray or brown on maturity. So in this diagram, lower diagram, you're seeing the thallus on which sporophyte is born. Sporophyte consists of foot, intercalary meristem, the upper part, capsule. In the diagram, you're seeing the spores are coming out, round spores. So it is somewhat complicated capsular part that we'll study now. The capsule. Capsule, it is the longest part, horn-like structure. It consists of center. At the center, columella is present. Center part is called as columella. This central solid region is columella, which is made up of 16 vertical rows of cells, one above the other. That is six. The cells are present. Many cells continuously. They are present. 16 such rows of cells are present, which gives support from the inner side. It extends from base to almost to the tip of the capsule. From the foot to tip, they are present. That is columella center part. Then surrounding the columella, you will see another region that is called as sporogenous tissue or sporogenous region. Sporogenous means that it has the capacity to produce spores. This is a mass of fertile spore forming cells surrounding the columella. So at the center columella is present. Surrounding the columella, sporogenous tissue is present. If you take the TS of that, you will see the columella at the center surrounded by sporogenous a ring, that is ring of cells. These sporogenous cells at the base of the capsule, it is single layered, the sporogenous region, single layered, and is called as archisporial tissue. This point you have to remember. At the base of the capsule, this is sporogenous tissue is just a single layer and it's called as archisporial tissue. It becomes 2 to 4 layer and develops into the diploid spore mother cells toward the upper side. So this sporogenous archisporial cells divides and redivides toward the upper side. It produces somewhat well-developed cells. They are called as spore mother cells, which are always diploid. Sporogenous tissue itself is diploid. Sporophyte is diploid. Toward the tip of the capsule, spore mother cells, they undergo meiosis, that is reduction, division, and produces haploid spores. So haploid spores are produced towards the upper side, in a mature sporophyte. Capsule wall. Surrounding the sporogenous tissue, you will see a layer that is known as wall of the capsule or capsule wall. It is the outermost wall of the capsule, which is 4 to 6 layers in thickness. This is not a single layer, 4 to 6 layers in its thickness. The outermost layer is called as epidermis and is interrupted by stomata. Here also stomata are present in the epidermis of the sporophyte. in the capsular region. So this stomata helps in gaseous exchange, and it also helps in photosynthesis, that is respiration as well as photosynthesis. That's why it is green in color in the beginning. The outermost layer is called as epidermis, which is interrupted by stomata. The inner layer consists of chlorenchymatous cells, that is chlorophyll containing cells, parenchymatous cells with chlorophyll, chlorenchymatous cells, and they are photosynthetic, means they help in photosynthesis. Next, life cycle. Life cycle of Anthoceros. Upper side, in the in this diagram you're seeing the toward the upper side, the lobes, that is Marcantia, not Marcantia, Anthoceros thallus, you're seeing with the lobes. On the lobes, if it is a monoecious, both are produced, Antheridia archegonia produced on the same plant body. If it is a dioecious, it is produced on two different thalli. So anyhow, in this diagram, on the same thalli, you're seeing toward the right hand side, you're seeing top right hand side, there is Antheridia development, that is Antheridia in the chamber. Two Antheridia are seen toward the left side of that, Archegonia present, Archegonia lower side egg cell, Antheridium, each Antheridium will produce so many anthrozoids which are biflagellate. They'll reach the Archegonia. They'll swim through neck cells by the disintegration of neck canal cells, ventacanal cells. Passage is formed due to that one of the antherozoid will enter into the Archegonia. There it fuses with the egg cell to form the zygote. This zygote divides and redivides and produces the many-celled sporophyte. Here, sporophyte, it consists of food, that is intercalary meristem, the middle part, and the upper one, capsule. In the capsule, intercalary cells that will divide and redivide and produces the sporophyte, that is sporogenous tissue. Each sporogenous tissue will produce spore mother cells. Spore mother cells will undergo reduction division to produce tetrads of spores after meiosis. Each spore, it germinates, and again in the favorable condition it will develop into the plant body of Anthoceros. So this is the life cycle in Anthoceros. Sporophyte, plant body is a gametophyte. On the gametophyte, sporophyte is developed, which is much visible distinctive feature, and that sporophyte will depend on gametophyte for its survival. So this is about Anthoceros. So with this,
Madam,
Anthocerosis, yes, sir.
Madam, thank you very much.
Thank you, sir.
It is a completely picturization about the morphology, anatomy, reproduction, life cycle, clear-cut idea about the uh, Anthoceros for all competitive exams. It is not only the academic side, but also it is most important, the all competitive exams. Each and every point, madams, you are covered in this topic. So thank you very much, madam. Tomorrow we will meet
Thank,
same time.
okay, sir.
Okay,
Thank you,
thank you.
sir. Okay.
Right.